Thank you, Don. Yeah, please. So we are going to, I'm so loud. I project when I teach, and so I think we'll fix the volume on here so I don't hurt your ears. Can everyone hear me okay? Am I too loud?
No.
No, okay. So as Don said, I feel like I'm very loud. I'm going to turn this down. So as Don said, I study plankton Foraminifera, and I do them in a variety of settings. I started out as a geologist, so that's my undergrad training is in geology. And then I went to grad school and I studied fossils, still sort of in the geology. I did climate reconstructions using fossil specimens that we during sort of last glacial maximum, so spanning modern to about 20,000 years ago. And during my postdoc, I was hired to sort of learn how to get this instrument up and running. And my postdoc advisor had a field season planned to go to Catalina Island and culture forams. And I got to culture this species right here, which is called a Neogloboquadrina deutertri. Well, I'm not going to quiz you later, but we call them forams for short also, so I'm going to use that word a lot. But I had the opportunity to grow them, and it completely changed my sort of research trajectory in general. And now I do lots of fun things that's very interdisciplinary. And I'm going to talk to you about how some species have puzzling geochemistry that's linked to their ecology, and how I figured this out using geochemistry and observations. So I'm going to start today's talk giving you sort of an overview of what forams are, because many of you don't probably study them in an ecology department, or if you're not in paleo looking at their fossil specimens. So forams are sand grain sized marine protists. When they grow, they make a test or a shell around their cytoplasm that's made of the mineral calcium carbonate. This is one here. Their shells are white, but this organism is very much orange because inside its cytoplasm are algal symbionts that are about 1 micron in size. The variety that I study are planktic, so that means that they live on the surface of the ocean. They're cosmopolitan in the ocean. This figure here, let's see if you can see my mouse right here, sort of shows you just their diversity. They tend to have the highest diversity where it's warm. So this looks like temperature, but this is actually sort of a heat map of where they're most common in the ocean. They live everywhere in the surface of the ocean. If you've swam in the surface of the ocean, you have swam with forams and you didn't even know it. They're also extraordinarily common in the fossil record. And so they have a fossil record that spans 180 million years for the plankton species. There's a benthic variety that lives in seafloor sediments and in estuaries and in reefs, and those have been around for 500 million years. So they have a very, very long fossil record, and this figure here is just demonstrating how researchers, this is from Watterson et al., used sediment samples that were aged to be about 20,000 years old, last glacial maximum, in their climate reconstruction. So they were studying their shells. So just a quick and dirty overview. I'm going to tell you about why they're so useful in the fossil record in just a few slides. In order to use them in the fossil record, we need to understand their ecology, right? And so that's part of what I do. My research is really interdisciplinary. I study their geochemistry, their ecology, their biomineralization. Can you hear me okay? Okay. Their biomineralization, and I do this in the modern and the fossil setting. Can everyone hear okay? Okay, awesome. So my research takes me to really fun places in the field. I get to go to, my field work takes me to Catalina Island. I think I've had something like 7 field seasons there in the last 15 years. I get to go on large ships off the coast of Oregon. I've been to Green Island just once. COVID shut down our 2nd field season, and I have not been back for that reason. When we collect samples, we collect them using plankton nets. So I collect both living and dead shells using plankton nets on big ships, on dive boats. I also collect samples from sediment traps that are just moored to the sea floor. These were out for 3 years, and now I have 3 years of data collected every 2 weeks that I'm correlating to surface conditions. And I also study sediment samples. So someone who I spoke with earlier today said, do the sediment samples just sort of look like mud? And it kind of depends on where you sample the sediment from. This is coastal Oregon. And so this is very muddy because there's lots of silt, there's lots of detritus, there's lots of fecal pellets, things that end up in here, lots of sand. These 2 layers right here are turbidite layers that happened during 2 earthquakes that disrupted the sediments. And so those are thick grains of essentially rock that were displaced and sort of landed there during some earthquakes. If you were to go to a study site that Don studies, for example, the sediments would look completely white, depending on the setting, because they're full almost 100% of just forams, white shells. So it kind of depends on the setting. So before I get into today's talk, I thought I would give you sort of a flavor of some of the other work that comes out of my lab. And this is going to be a super highlight overview, but I'm happy to answer questions after. One of my students, Kelsey Lane, who's now my colleague, she studied the impact of marine heat waves on foram ecology. So forams live in communities. They thrive in specific temperature conditions that are sort of keyed into the species. And off the coast of Oregon in 2015, we had a marine heat wave. It was actually 2014 to 2015. Marine heat wave is basically when the ocean warms above background for an extended period of time. This one was very warm. It changed the ocean surface temperature for almost, I think it was something like 750 days. And this is a map of temperatures during 2018. The cool colors are, the anomaly is like slightly cooler to normal. This is 2015. And the take-home point here is really that when it's cold, the species that are present are cold water species. So you can think of the blues and the purples, the species names are kind of irrelevant. The species that thrive when it's cold are cold water species. And during the marine heat wave in 2015, the blue species, the ones that live in the polar regions, completely go away. And we have an influx of this pink color here, which signifies tropical forams. So we had tropical forams living off the coast of Oregon. 2 species, one of them had never been seen in Oregon. We've actually now seen 2 that have never been seen that we know of off the coast of Oregon. And so this is sort of what's to come as climate changes. We will see more of these species. Other species also changed in response. So for example, euphausids and copepods that were tropical species came into the region during this period of time. Kelsey's work also included microbiome association. So she's looking at the bacteria that live inside the forams. And she's also been doing work with morphometrics and genotyping. And those papers are either in prep or published. But what I'm mostly going to talk about today is culture experiments. So I grow forams a lot to understand what makes them tick, right? We want to understand what controls the composition of their shells. And we do this by growing them in really highly controlled conditions where we might keep everything constant except temperature, or everything constant except the pH. And so what I've been doing for the last couple of years is growing them in high temperature and high CO2 conditions that will sort of mimic what we're going to be at year 2100 to see what's going to happen to their shells. So they will grow thinner. This is something that we know, that under high CO2 conditions, they have a harder time building their shells. But part of this work is also to understand what genes are responsible for calcification and to identify the proteins that play a role in this process. And so this species that I have here, to sort of orient you to what you're looking at, inside this, it looks like a bubble capsule, but it's not a bubble capsule. I'm going to sort of walk you through this. This is the juvenile on the inside. And so this is probably about 150 microns, maybe. And when this foram grows its adult shell, it adds this chamber on the outside of it. And so that is a thin layer of an organic membrane that it sort of, the envelope of the organic membrane comes out, and that's what it templates its calcite onto. And what we were trying to do is capture this exact moment that this was happening, and then we killed the forams. We put them in a DNA buffer. And what we're hoping to do is figure out what genes are responsible for the biomineralization to happen. So a lot of foram torture is essentially what I do in the lab. So that's some of the work that I've been doing. And then I wanted to just also sort of the foram species that's there. This is called a spinose foram. And so what looks like little rays of light coming off the shell, those are actually individual calcite crystals. The little dots around it are algal symbionts. So all these little dots are single-celled dinoflagellate algae. They're the same genus as the symbiodinium that are in corals. And so they help the forams calcify by enhancing the carbon system around their shells. And just to give you perspective, this is one of the larger species. It can grow about as large as 2 millimeters. And when we have them in culture, we feed them day-old monkey brine shrimp. So we buy them from Amazon, very, very technical. And we pipette day-old brine shrimp into their rhizopodial network, and that's how they eat. And so they require symbionts to grow and thrive, but they also require food. And so if we starve them, they die. If we overfeed them, they die faster. So they literally require being fed. And that little artemia, the more it moves, it gets entangled in its rhizopodia, and it's going to pull that little artemia up against its shell and dissolve it. And then it doesn't eat the carapace; it eats the stuff on the inside. So lots of artemia torture as well when we're in the field. But what my talk is going to be about today, the focus of my talk, is that I want to discuss the differences between two very specific types of forams. The spinose variety are the ones that have spines. The non-spinose variety, black spines. So that sounds very simple. There are about 90 species, about 55 or so that we have identified morphologically, and about 50-50 have spines and don't have spines. And this fundamentally changes how they behave in the ocean, what they eat, and it changes their geochemistry. And for a long time, folks didn't understand what was causing them to have different geochemistry. And some of my work and my collaborators have sort of solved this puzzle. And so ecology matters. And so that's sort of what I'm going to walk you through today. The spinose forams, that little fluff behind it, that's a piece of marine snow or particulate organic matter. And so if you've swam in the ocean, or if you've seen movies where you see sort of sparkles of light in the ocean, some of those are organisms in the ocean, but some of it is just sort of the detritus. As algae and organisms die in the ocean, they decompose, and their decomposed matter is very sticky, and it makes particles of marine snow. And that's what that foram is stuck to, and this plays a very, very large role in the life of the non-spinose forams, and I'm going to sort of walk you through that today. I also welcome questions. And so if I get confusing or if I say an acronym, I tend to speak in jargon. This is something that I told the folks here beforehand. And so if I say an acronym you're not familiar with, please feel free to interrupt me. So in terms of like, why do we study forams and why do we care where they live, right? They're very small. They're hard to work with. They're incredibly useful in paleoclimate research because every species has a very well-defined ecological niche. So I'm going to sort of walk you through this. This is the spinose and non-spinose. I have 3 S's next to these. These are 3 spinose species. I have them colored because I'm a nerd and they have orange algal symbionts. The spinose forams are primarily shallow dwellers. 70% of the ones that we have studied so far for the presence or absence of symbionts have them. So they're going to be sort of restricted to the photic zone because of that need, their algal symbionts need to have sunlight. These are also really widely used in paleoclimate research, and sort of for 2 reasons. The first one is that they've been really widely used in culture experiments. They're very amenable to being grown in culture. We can sort of torture them, put them in, break their spines, get them to regrow their spines. They're very happy in culture, and so we just know more about what makes them tick. So that's sort of the first reason. But also they live at the surface of the ocean. They really do. Some of the species live right at the surface. And so for researchers who want to reconstruct the surface ocean conditions, these forams are the most important. They're the ones that are sort of living in the parts of the ocean that talk to the atmosphere. If you want to understand paleo pH, for instance, in the surface of the ocean, you're going to use a foram that lives in the surface of the ocean. But there's some power to having information from all of the different species. The non-spinose forams tend to live deeper in the water column. Some of them have algal symbionts. About 50% of the ones that have been investigated so far have symbionts or active chlorophyll within their cytoplasm. And so that restricts some of them to the photic zone. And then there are some varieties that live completely out of the photic zone and don't have algal symbionts at all. What makes this really useful for paleo is, number one, they have a well-defined niche. Number 2, if we want to be able to understand, changes in the thermocline in the past, we can do reconstructions with multiple species that occupy different ecological niches, and we can reconstruct the entire water column using information that's sort of stored in their shells. So there's some power into sort of having that information. They are underutilized. The non-spinose varieties, I would argue, are a little bit underutilized in paleo. It's not to say that they're not used, but we often take what we know about the spinose variety and make assumptions that the non-spinose species do the same thing. And I'm going to show you in my talk that that's probably incorrect. They're also underused in culture experiments simply because they're so much harder to grow. When I started my postdoc, there was one published study in 2011 on the non-Spinose species. And so I really sort of went into the culture field, not having any idea. I think my first 3 weeks in the field, I took in plankton samples and they died overnight, every night. Couldn't get any to survive. So it took me a field season to figure it out. So they're not widely used. We actually changed this. They're actually very happy in culture. We figured out how to get them to grow and thrive in the culture setting. So when I say that they have well-defined ecological niches, you might be thinking to yourself, well, how do we know? How do we know where they live? So what we've done in our field, and folks by many decades before me have been capturing them in plankton nets. And often they will use something called like a ring net is just going to sample the entire water column. But there are nets that are called multi-nets or multiple open-closed net environmental sampling systems, MOCNES. So those things can go to like 1,000 meters. And then you bring it up to the water column to maybe 500 meters and close that net. Bring it up to another, couple hundred meters and close that net. So you're able to get discrete samples from the water column. And then you can simply see what species are present. So the upside to that is you can sample the entire water column. The downside is you're going to get samples in there that died from shallower depths. And so the second part is that we can measure their isotopes. And so this is a highly cartoonized sort of picture of seawater has an oxygen isotope gradient, and I can measure the oxygen isotopes of the species, and I can sort of reconstruct where they're living. They're very good geochemists, and most of them record the oxygen isotopic composition of the water that they're growing in. So we have sort of two pieces that help us figure out where they're living. Makes it very useful for paleoclimate. And so, as I'd said, they are very good geochemists. This is what makes them powerful. Don mentioned this in the beginning, that their chemistry reflects their growth conditions. And so these two live forams here, those are photographs that I took. Those are photographs of what their fossil specimens look like on the seafloor. I mentioned that they're made of the mineral calcite, but when they grow, they incorporate a whole bunch of trace elements into their shell. And the quantity of those elements and the isotopic composition of those elements varies with growth conditions. So if we've measured magnesium, boron, barium, strontium, cadmium, like if it can be incorporated into the crystal lattice and we can measure it, some scientist is trying to figure out how we can exploit that data for paleoclimate. And so we can also measure the isotopes, things like oxygen and boron and carbon isotopes, and correlate that data to their growth conditions. So if you're unfamiliar with this, I would like to walk you through sort of how one of these work. One of the most widely used elemental ratios in forams is the magnesium to calcium ratio. This is what's used as a paleothermometer. And so we can't, I like to call forams sort of like time travelers. They're time machines. We can't go into the past and measure the ocean temperature, but we can go into the past and measure the magnesium in their fossil shells. And the way that this works is their magnesium-calcium ratios vary with temperature. And so this is a multi-species calibration study where scientists took a series of different species. They determined the temperature at which they calcified, measured the magnesium-calcium ratios in their shell, and demonstrated that there's this nice exponential relationship between the temperature that they grew at and the magnesium-calcium ratios in their shells. And this has been done in a variety of ways, sediment traps, core top studies, and this can also be done in the laboratory setting. So I've put these little arrows down there to sort of demonstrate. We might put forams in culture at 14, 20, and 26 degrees, and we keep everything constant, their light, their food, salinity, pH, modify the temperature only, and then measure the magnesium, and we get something similar. So this is a sort of very highly cartoonized version, but fairly consistent, that the magnesium-calcium ratios change by about 7 to 10% per degree C. Now, there are some secondary factors, pH and salinity play a role, but predominantly it's temperature. Very widely used temperature calibration. Makes them very useful for paleoclimate. I won't walk through these other ones, but folks like Don use the boron-calcium system or boron isotopes for reconstructing paleo pH, or information about the dissolved inorganic carbon system in the ocean. The power to some of this is that you can measure boron calcium and magnesium calcium in the same sample, and you can reconstruct temperature and pH from the same set of samples. What I'm going to talk about today is barium. So barium is a little bit of a puzzle, or was a puzzle in forams. The incorporation in species is inconsistent. It's not predictive. And it's been relatively poorly constrained in the non-Spinose species for quite some time. And I'm going to sort of talk you through how we figured out what we think is causing the barium to vary in the species. And that really came from culture research and observations. And so when I mentioned that magnesium is a really good temperature proxy, I want to sort of show you what well-behaved sort of paleoproxy data look like, and even in the modern setting, that things behave like we expect. When I was in Taiwan in 2019, I collected a bunch of samples from the plankton tow sample, and I measured the magnesium calcium and the barium calcium ratios in their shells. And this is a figure of what the magnesium calcium ratios look like, just to sort of keep you cued into where they're living. 3 of them are Spinose species. They have the S's next to them. And the pink one up there, the NS is missing, but that's the non-Spinose foram here. Their depths, right? One of them is a surface dweller. It has the highest magnesium because it's calcifying in the warmest water. And as the depth habitats decrease, so does the magnesium calcium. This is exactly what we would predict, and it works very well. This is what the barium data look like. So when I first generated this data, it's sort of like one of these things is not like the other, right? It's very, very different. They're living in the same water column. The barium calcium ratios don't vary over the depth of calcification. Salinity doesn't vary. pH doesn't vary. The oxygen doesn't vary. They're all growing in the same water. The difference is that that one doesn't have spines. All of them have algal symbionts. So not only is there a difference in terms of the magnitude, but these are all individual. This is a box and whisker plot of like maybe 20 shells. They have very, very different barium calcium from specimen to specimen. So something else is happening. And the reason that I put manganese up there in parentheses is I could swap this plot out and just put manganese calcium ratios, and I see the same thing. There's no manganese in the Spinose forams, and the manganese is very high and variable in the non-Spinose species. So they're doing something different. In the fossil specimens, these 2 ratios are attributed to diagenesis. Forams shouldn't have manganese in them, for one. We have an oxygenated ocean. There isn't enough manganese in the ocean to explain this. So it's already a little bit puzzling that these are plankton tow samples with such high manganese and such high barium. And it's not just the samples from here. This is non-Spinose data. So on the x-axis, the species names are almost irrelevant. What really matters here is that the first 5 are the non-Spinose species, and the last 3 are the Spinose forams. So this is data from Catalina Island, Taiwan, off the coast of Oregon. And the barium calcium ratios are almost always high. And if I replace this with manganese, I would see the same thing. So fundamentally, these forams are doing something completely different. And I'm going to highlight this one because this data is going to matter in a couple of slides. So this is data that I collected in Catalina Island in 2015. So this is sort of insights from the plankton record. They're fundamentally doing something different. So from a historical perspective, back in the early 90s, there were some researchers at MIT, David Lee and Ed Boyle, and they thought, I wonder if we could use barium in fossils to reconstruct the barium composition of seawater. In order to do this, they have to understand the mechanisms of barium incorporation. So they collected a bunch of samples from the seafloor. Samples from the seafloor are dirty, right? You have to clean them. We have to get rid of clay, things that can also cause the barium to be elevated. The forams are not, to get the primary signal, you have to get rid of everything else. So their first step, this is a plot here with 4 species on the bottoms. 3 of them are Spinose and 1 is non-Spinose. And they did 2 cleaning steps for the shells. This is fairly common for all of us that do fossil work. First thing to do is we need to get rid of the clay contamination. And to do that, we put them in distilled water. We ultrasonicate the shells. Sometimes we crack them open to get stuff out of the inside, do a bunch of rinses, try to get rid of the detritus that way. They did that, measured the shells, and they found that they still had highly elevated barium. So the hashed marks are samples where all they did was rinse them. Then they did a series of additional cleaning steps that are very, very corrosive to calcite and also remove metal oxides. So if there's any diagenesis in the water column that can make the manganese go up, or if there's barite contamination, which can happen in the seafloor, this is going to clean that off. And so their first round, after they applied these really highly corrosive cleaning steps, they were able to get the ratios down to about 1, less than 1, the little red arrow there. So when they did that for all of their samples, before they started, they had 33 samples that were spinose that had elevated barium, and they had 21 samples that were non-spinose that had elevated barium. They applied the rinses, which were insufficient. They applied these cleaning steps that were meant to get rid of all of the stuff that they didn't want, but the cleaning turned out to be very, very inconsistent between the two. What they basically found was that they couldn't get rid of the elevated barium in the non-spinose forams, the ones that don't have spines. These cleaning steps got rid of all of the barium in the spinose forams. Just 3 out of the 33 samples, 9% still had elevated barium. But out of the non-spinose forams, the barium was still elevated in 62%, right? So they're trying to figure out why. They cleaned them. All of these samples are where they cleaned the samples. And what we're showing here is the post-cleaning sample recovery. So even if they had like almost no shell left, all of the metal oxides are gone. They're still having ratios that are on the order of like, 2 to 8. So this was a puzzle to them. And they thought, okay, is this still contamination when they publish these papers? Maybe they just have more barium in their shells to begin with, right? What's causing this? So I just showed you that in the plankton they do, but they're looking at fossils back in like the 90s. So they already knew that this was a puzzle. So the first steps for solving this puzzle are, of course, to grow forams in the laboratory setting. And so David Lee and the person who was the previous author on the fossil work and my postdoc advisor, Howie Spiro, in the early 90s, did some culture experiments. And then in 2011, Beryl Honish did an additional one. What they did is establish the partitioning of barium into the spinose forams. Growing the forams in the lab, so I'll sort of walk you through what this plot is. These are samples that are grown in culture. They took seawater from the study location, added some barium chloride to the seawater. They had 5 experiments, ambient seawater at the bottom, and then they increased the barium calcium ratios in the seawater, grow the forams in the seawater, measure the barium calcium ratios in the shells, get this wonderful linear relationship. This basically says, hey, when forams grow in the ocean, they're recording the barium composition of the seawater that they're growing in. So this is sort of like the first step in understanding barium incorporation. So what this also meant was that when the forams are growing in ambient seawater, they should have low barium, right? So it became very, very hard then to explain this, right? It still brings us back to this puzzle. They're showing the spinose forams incorporate barium as a function of seawater. They should have low ratios. So naturally the question becomes, maybe they just have a higher affinity for barium. So their partitioning for barium is greater. The slope is like this, like off the charts high. So what needs to be done is we need to repeat this experiment, but using non-spinose forams. And so that sort of enters my work in 2015. So in 2015, we're on Catalina Island and we collected some N-deuteritri. This is the non-spinose foram that I've shown the picture of. And we put them in seawater. We essentially repeated this experiment, 3 different levels, ambient conditions, 2.5 times ambient and 5 times ambient. But this time I added an 87 strontium isotope label to the seawater because I'm going to analyze them using something called a laser where I can identify just the calcite that grew in culture. So the way that this works, this is the little tiny foram that's about the size of a grain of sand, and I can take a laser. Don has one of these if you want to know how they work. So laser ablation ICP-MS, the laser spots are about anywhere from 25 to 65 microns. They pulse. Every time they pulse, they remove a thin layer of calcite. That calcite gets sent to a mass spec. And the parts that grew first are on the outside. I'm going to identify the cultured calcite using the strontium calcium label. So what I should expect to see is any calcite that grew in culture is going to have this 87 label. Anything that grew out in the ocean is not going to have the label, and partially grown shells would have this transition between the two. Now, any chamber that grew 100% in culture would be fully labeled, right? But this allows me to sort of increase the number of specimens that I can analyze because I can just take the calcite that grew, that got added. So this is an example of what a laser ablation profile looks like. So to sort of walk you through this, the ablation time is at the bottom. So this is going from the outside of the shell to the inside of the shell. The label is at the top in blue. The put 0.34 is the 3 times ambient 87. So this label, this calcite grew in culture, and then you can see it sort of plummet, and that thin little veneer at the end, that part of the calcite grew out in the open ocean. So I can take that bar, that labeled calcite, and I can quantify what grew in the lab. And so we did this at those three levels, and this is my calibration equation that was published in 2018 for establishing the partitioning into the non-spinose species. Looks pretty similar, right? It's actually statistically the same. They have the same partitioning, and deuteritry that's growing in the open ocean should be incorporating barium calcium of the seawater that it's growing in. So in the open ocean, they should have ratios on the order of about 1, less than 1. So in 2019, we did a second species, the one that I showed on that first slide with the really high barium. If I put those in culture, they're also recording ambient conditions. And in 2023, because I needed to do this on yet another species, I did this experiment off the coast of Oregon with a polar non-spinose foram. And we're still working up the data, but we have the same exact relationship. So 6 species grown in culture, 3 of them spinose, 3 of them non-spinose. They are recording the conditions that they're growing in. The punchline is that forams grown in seawater with ambient barium should have barium calcium ratios on the order of about 1. So how do we reconcile this? Right, if I put them in, if I take them from the open ocean setting, put them in culture, they record the water that we're using in culture. This is the water that we take from the same place that we collected them from. The 3 spinose species at the bottom here, and the 5 non-spinose forams, the asterisks are one that we've grown in culture. It becomes really hard to reconcile the data from the ocean with what we're seeing in culture. And so this is where observations ended up playing a huge, huge role in this story. So we think that the forams are living in particulate organic matter out in the open ocean. This is part of their ecology. So the story behind this discovery is kind of like serendipity. In 2015, that picture on in the end and the one in the middle, this is a tropical foram. In 2015, we were on Catalina Island during that peak marine heat wave that hit the West Coast. The water conditions, we had a running list of tropical species on the refrigerator in the lab that had like never been seen. This species is not seen on Catalina Island, the one in the middle. There were so few forams in our net tows that I seriously thought my field season was sunk. And I started taking little particles. It's even hard to see the foram in the one on the end. Took a paintbrush and I started peeling the marine particles apart, and I found forams in them. So I thought, oh my gosh, are they living in marine snow? Okay, but we're collecting them in plankton nets, right? They're very, very sticky. They have cytoplasm all over them. Are they just enveloping themselves in marine snow in that process, right? So we had to do a little bit more work to prove this idea. But why is this a potential microhabitat for them? If you're not familiar with what particulate organic matter is, I sort of mentioned that it's the breakdown of things like algae when they die. It's very sticky. It's full of like polysaccharides and things that get stuck together. Inside marine snow particles are a very unique habitat, like very unique environment. There are researchers in like the 80s and 90s that put microelectrodes inside marine snow particles, and they were able to show that marine particles have low and variable pH, like 0.2, 0.25 pH units lower than the ocean. This is a significant difference in ocean pH. That's like the kind that we would expect, you know, when the ocean changes to 2100 or last glacial maximum. These are very, very large changes in pH. You can have anoxic conditions inside marine snow particles, no oxygen, right? So that's going to change the microhabitat that the forams are living in. This is where barite forms in the ocean. So barite does not freely form out in the open ocean. It forms inside marine snow particles. The ocean is undersaturated with respect to the mineral barite. So when it forms, it wants to dissolve. This is the happy spot for barite, where it can grow. So we know that the barite of the seawater inside these particles has to be really elevated. What does this mean in terms of the forams geochemistry? They're all living in their own little home. right? So every single foram is going to record a different barium calcium ratio. This is what's driving the interspecimen variability in our plankton nets, because they all have their own little house. It also is going to drive something that I didn't show today, but when I take that laser data and if I analyze the shells from the inside out, the shells on the inside are nice and flat, right? The shells on the outside are bumpy because of their pores. But if we analyze them from the inside out, we can see sort of intra-shell trace metal variability, and I can see these diurnal bands in the barium. I can see these bands in the manganese. They're respiring. Many of these have algal symbionts. They're going to drive that pH and that oxygen up and down daily, and they're going to record that in their shell. So really neat mechanism for explaining this. The last piece of the puzzle was, do they do this on purpose, right? So how do you get this? How do we get this observation? Do they wrap themselves in marine snow? Benthic forams do. Benthic forams make a feeding cyst. They live in sediment and they take their little rhizopodia and they put stuff around them in the sediment. So in I'm going to pause this for a second before I get started. 2021, I was on a ship off the coast of Oregon and we collected some non-Spinose forams and we fed them marine snow. And I'm going to sort of pause this and walk you through it. They will readily embed themselves in marine snow. And so this is a species called Pachyderma. It's the polar species. We don't know what causes its color. This one does not have algal symbionts. Playback speed is about 20 times. And I'm going to hit pause for a second. The recording ended or started about 5 times after I pipetted a piece of marine snow onto its rhizopodial network, and I did it right about here. This is where I put a piece of marine snow. Now, when you're looking at this, it looks like these sort of rays of spines are emanating from the shell. Those are not spines. That's an extension of its rhizopodia. So when we grow these forams, they have a behavior in culture. We call it going benthic. So we capture the non-Spinose forams. They sink to the bottom of the jar. We grow them in falcon flasks. They're optically clear on the bottom. They stick. They stick so well that I could literally like throw one across the room and they're not gonna, they don't let go. So they're very stuck. So this is just its rhizopodia sticking out. No spines. Okay, so I want you to see what happens here. It's taking the piece of marine snow and it's pulling it in and it's in sort of enveloping itself in the marine snow particle. And so we think that this is behavior that they do, continues to do this. And then the next day, I took some observations, and the shaking back and forth is because we had about a 13-foot swell. It was not very fun to look down a microscope. So this was the following day. And this was probably one of the most exciting days that I've had in all of culturing, and I'll explain why. So this is the particulate. I don't know if you can see the shell. You can try to sort of draw around it. Here's the shell. This is a ball of particulate material, the marine snow that it has now pulled up against its shell. And those are not spines. Those are still its rhizopodia, but this time it's floating. We don't get these things to float in culture. So it's enveloped its shell in marine snow and it let go of the culture jar. So that was pretty exciting. I think I scared people on the boat, to be honest. I was pretty excited. And so after that, it was attached, and we have gotten this to happen repeatedly. We did it with 3 different species. They are picky. They want it to be a piece of fluffy marine snow. If I feed it something that doesn't quite look like this, they don't take it in. And some of my collaborators have repeated these experiments. They really like to have the particulates. And it was not, I've had the question whether or not they were swimming. This is not swimming. This is, again, we had that 15-foot swell or whatever. I was having such a hard time keeping it under my camera and keeping it in focus because it kept moving. So exciting. So this was very big for me. You can probably like feel my excitement. I was very excited when this happened. So the question then became like, can we exploit this for paleo, right? So some of my collaborators have repeated these experiments. They've done this with tropical forams in the Gulf of Mexico. They've done experiments trying to feed. One of them is this Truncatulinoides in the Gulf of Mexico. That they tried to feed them. We typically feed them Artemia that we hatch for one day and then freeze. We can't feed them live Artemia. They don't take in the flapping live Artemia. The truncats would not even take the Artemia, wouldn't even accept it as food, and they took the marine snow particles. So this paper is hopefully accepted. It was accepted with minor revisions. She has a second paper in 2022. So how can we exploit this for paleo? This is the idea that we sort of presented, is that if non-spinose forams are relying on particulates for food, substrate, floating, this is going to have a profound effect on the trace elements. And because of the redox chemistry in there, can we use this to reconstruct paleo productivity? So I have a couple of slides that I'll walk you through on this. The first piece was, if I take modern forams and I measure the barium calcium ratios in their shell, does it vary with export productivity measurements in the ocean? And it does. So what I'm showing you here is on the x-axis, this is modeled export productivity measurements. This is the moles of carbon per square meter per year, plotted against the barium calcium ratios of some shells that we took from core top samples, so the young samples that are on the seafloor. We also took them from sediment traps in the equatorial Pacific. We measured them on a laser, and we measured them also in solution. So two different types of analyses. And we have a nice relationship. It's quite noisy. This would benefit from a larger study. I'm working on the proposal for this, but essentially we get a fairly nice relationship between the two. Barium calcium ratios vary with productivity. How does this look in the fossil record? We actually went to the fossil record first before we went to the modern. But so in the ocean today, particulate organic matter or marine snow formation varies with productivity. So if you have high productivity in the ocean, there's going to be more marine snow. And not only is there going to be more marine snow, the particle sizes are really different. So you can have large particles, small particles. This is work that Phoebe Lamb has done. So high productivity, high palm production, elevated barium, high variability from specimen to specimen, because you have lots of different sizes and shapes of particles that they can be living in. During periods of time in the past when there's low productivity, and this also happens in the modern ocean, if you don't have a lot of productivity, you don't have very much marine snow. You're not going to have a lot of variability in particle sizes. They're all going to be small. So they are going to have lower barium calcium ratios and reduced population variability. So it's just going to shrink those box and whisker plots for, if we measure an entire system or an entire sample individually. So we can take a down core record and interpret it within this framework. So this is exactly what we did. I had a graduate student named Teresa Fritzendris. We wrote a proposal, and basically we picked two locations in the Pacific using sediment cores from the Eastern Equatorial Pacific and the Ontong Java Plateau. And we chose these two regions purposefully because today they have very different productivity. So we wanted to be under a region, for one, that has different productivity. We also chose this location because so many researchers have already investigated paleo productivity using these two sites, using different methods. So this is sort of like a case study. Proof is in the pudding. What do we find compared to existing studies? So we also did two different types of analyses. Laser ablation, I get a bunch of individuals that I can measure individually and get sort of a population statistic, range, box and whisker plots. But if anyone has ever done individual analyses of anything, that is very time consuming. And so the answer really is to do solution analyses where we put them all together. So we did both. So I'd like to walk you through these results, and feel free to ask questions on this for clarity. I know I've already been talking for over 40 minutes. So we did this in 3 time slices for the individual laser ablation specimens. We did samples from the Holocene, the deglaciation, and the last glacial maximum. We have radiocarbon dates on these. These are very well-used, well-loved cores. We did this through the time slices are the laser data, and then in between the smaller dots, and also the error bars are based on solution analyses where we took 30 specimens and combined them together. We did 3 species. So the 2 species, the blue and the green, are non-spinose forams. And the red is the spinose foram that's recording seawater. So the first take-home message is the species that faithfully records the barium calcium ratios of seawater is doing exactly that. This one down here. So this was sort of like our control. This one should only have ratios on the order of about 1 to 2, and that's exactly what it has. These forams are the non-spinose species that we think should be recording paleo productivity. And there is a well-known and well-described paleo productivity spike that happened during the deglaciation in both basins. And that's what is described by our data here. So during the Holocene and the last glacial maximum, they have sort of similar productivity. During the deglaciation, productivity was elevated. This has been shown in other proxies to be the case. And so we think we have another way of sort of reconstructing paleoproductivity using a proxy carrier that people measure barium in anyway. So it sort of adds an additional piece of data that folks can use going after this. The only other thing that I want to mention, I know there are not a lot of folks in here that like study forams, but cleaning is extraordinarily important. I showed that data from the Lee and Boyle study. If you over clean the shells, you get rid of this signal. It actually preferentially removes parts of the shells that have high barium. And so cleaning matters. And this came out of Teresa generated some data and came in and said, my data are bad. And I said, your data aren't bad. We just have to figure out what they mean. Prompted a cleaning study, and we learned that she had overcleaned her shells and removed the primary signal. So cleaning ends up being really, really important. And so, yeah, we think that we have a new mechanism that explains the barium in these species. They're fundamentally doing something different because they're living in marine snow. This also, circling back a bit, since I didn't say this also explains the manganese. So I don't know, when I mentioned that the manganese can be high, manganese should not be high in planktic forams. The seawater doesn't have enough manganese in it. They're getting the manganese by being in a setting that has low oxygen, that allows the manganese to stay in solution. That's where they're getting it from. They're living in a low oxygen environment. And so I think that's all I have. So my conclusions for this are that understanding the non-spinose geochemistry has really been key for understanding where they're living, understanding their ecology, and really being able to use them in the paleo record. This is also important for understanding what's going to happen in a future ocean, for understanding where they might grow, evolve, and thrive. And we have now a potential proxy for paleoproductivity, and there are some potential implications or potential problems for their use. Is that we've been making a lot of interpretations about what controls their geochemistry based on the Spinose forams. The magnesium is impacted by this environment. So they might not be the best proxy for reconstructing temperature if their magnesium is incorporated by this or is impacted by this environment. I have a paper that shows this nice spike in magnesium during the deglaciation. In those same cores, it's probably driven by this, it's not temperature. So that's one potential issue. And a second one is that non-spinose forams are potentially, boron geochemistry might be affected by this because they're living in a low pH environment. And so for folks interested in looking at boron for pH, this complicates that utility. And I think there are some folks looking at this already. And so things that I would like to do more about this, I'm done doing culture experiments on this. I don't think I need to do another partition experiment. However, what we are trying to do is grow them in labeled particulate organic matter because I want to know where the carbon goes. I want to know what are they incorporating into their shell. And so we can do that by tracer experiments, by growing them in like marine snow that we make and label, and then follow that with like TEM and nanosims type of analyses. And so I'm happy to answer questions. Thanks. Yes.
Awesome talk. Question about the evolutionary history of this trait and whether or not that might impact your analysis. So first of all, are these non-spinose forms, are they a monophyletic group? Like, is this a trait that you think led to their adaptive radiation ability to use?
Oh, that's a good question. So one of the researchers that I've spoken to about this is Ellen Thomas, who looks more at the foram sort of evolutionary history. The non-spinose forams are more genetically related to the benthics. And so this might be behavior as they evolved all of them evolved from the benthics. But if you look at the timing of their evolution, the benthics are actually more genetically related. So this might be part of the reason why. I don't, but I do know researchers who are sort of probing this relationship in the past. The trick is cleaning them so if they are diagenetically altered, some of the diagenesis that can happen in the pore water can make barium go up. So that impacts this. So I think cleaning is key, and some careful analyses need to be done. Like things like nanosims, I think are useful. Microprobe is not that useful because the limited detection is not great, but nanosims, I think, can get at like, where is the barium? Sorry, I keep saying the word like. I don't know what's happening to me. So certainly there are non-spinose forams in the past, and we might be able to look at productivity in deep time if we understand where they're calcifying. Yeah. Yes.
That's about carbon isotopes.
Oh, yeah.
So if they're full of barium.
Yep.
It's coming from barite that's forming in these particular organic matter particles.
Yep.
Where it's low pH, low oxygen. That's because of respiration of that organic matter, right?
Yeah.
So there should be really, really low delta C13 the dissolved in organic carbon in that environment. So in general, do those non-spinose species have lower carbon isotopes, and on an individual level, like, is there a correlation between individuals that have high barite or
Yeah.
high barium, say, and low delta C13?
No. So you're absolutely right. So first of all, if they're eating detritus, like organic matter, and incorporating that into their shells, they should have very low carbon isotopes. That doesn't behave in the direction that you would expect. There are offsets though. So we have some really great data that we're still trying to interpret. I think part of it is that where are the forams getting their carbon, and what part of that carbon ends up in their shell versus the organic membranes. So we know when they eat, nitrogen isotopes are impacted by the food. So we actually just have a paper coming out that shows if you vary the nitrogen isotopic composition of their food, it ends up changing the nitrogen isotopic composition of the organic membranes. And I think that we'll find that with the carbon isotopes, that it's changing. It's not the same pathway. They vacuolize seawater. They're getting the carbon from the seawater. It's really complicated. So part of what we wrote in that proposal that's in review right now is to track, basically we're going to grow them in elevated carbon so that we can track it and see where it's going. Yeah, that's a really good question. It doesn't behave like you would expect. Yep.
It seems like another complication. Even though they live in these little packets of organic matter, they also have symbionts, right?
Yep.
They're doing photosynthesis. And so the balance of photosynthesis and respiration,
yeah,
might be totally different in, the pool DIC that they're calcifying from.
yes. So it's going to significantly alter the microhabitat right up against their shell where they're potentially taking in that seawater. And this is some of the banding that we see in intra-shell. So inside the shells, as I ablate them from the inside out, very highly elevated barium, very highly elevated zinc and manganese. And this is happening in that microhabitat in a species that has symbionts. So yeah, a very, very complicated environment. Yep. I think there's still a lot we have to learn, I think, on that. Yeah. That's a good question. I think we definitely expect them to migrate. They're going to migrate north, right, into warmer water. The species that are going to lose are the polar species. So they're anticipating that diversity is going to decrease in the tropics, probably increase in the polar regions. Paper just came out saying that some forams, that migration isn't enough and that some forams are just not going to make it. And I totally disagree with that study. Happy to talk about why. So will we see new species? I don't know if they're going to radiate that fast. Forams are very robust, though, to climate change. I don't know if you agree. I mean, we've gone through massive extinctions. 70% of the benthic forams went extinct. 70%, Don, is that right, during the PETM? Yeah, 70 went extinct. And so after that, there's usually some sort of irradiation. They sometimes decrease in size. People have thought that they lose their symbiosis during periods of climate shuffle. I don't know that I buy that. If we make it so that the forams can't so that their symbionts don't photosynthesize, we can put them in an algal photosynthesis inhibitor called DCMU. Forams stop growing. They literally need their symbionts. The symbionts don't die, they just can't photosynthesize. Forams stop growing. If we put them in the dark, they stop growing. So I don't know that we're going to actually witness a radiation. I think we'll witness more of like extinctions, species sort of losing out. Any other questions? Yeah.
Their evolution.
Oh, that's a good question. I don't think I have the answer to that. I know that they have done some work to sort of kill off the symbionts and then reinfect them with symbionts. There's a big argument in our field on whether or not they're obligate versus facultative. Like, do they have to have the symbionts? I think if they have the symbionts, then they have to have them. But I don't know. I don't know enough about the evolutionary, like, history of the symbionts to answer that question. I think I saw a hand up. Yeah, so there are features that folks look for in the shells themselves to determine whether or not shells are completely diagenetically altered or if they sort of retain some of their, I don't know, fabric. Don would probably know this better than me because I don't do deep time forams. But there is intra-shell trace element variability that when they diagenetically recrystallize, that goes away. So their isotopes change. So there are some things that we can look at and go, oh, those are not ones that we want to use for paleo. So we do kind of work to find sites that are not diagenetically altered, but it's pretty hard. The farther you go back in time, I think the more difficult it gets. I sort of stay in like the last 100,000 years for that reason. Going into deeper time is a lot harder, a little bit more challenging, but worth it because we learn a lot about the past. Any other questions? Thanks, everybody.